Welcome back. So in this video, we'll talk a little bit about the basics of collecting data, talking about variables, and talking about experiments. So we'll cover first what statistics is, why you're taking this class or why you're watching these videos. We'll talk about how we can describe a data set. So we'll talk about variables, observations, and then types of variables. So first, let's talk about statistics. What is it? Well, statistics is the study of methods that can be used to describe and measure aspects of some population from a sample. So given data that we've collected, how can we use that data to infer something or learn something about a population that we wish to study? Some other definitions of statistics. We have the art and science of learning from data. Statistics is a science in that there are some procedural steps that we'll want to follow. It's also an art in that there is a lot of subjective judgment in how we choose which procedures to actually use. It's also the study of how to collect, analyze, and draw conclusions from data. A lot of people think of statistics as just being that step at the end where you run some analysis, you get some number, that number tells you what decision to make, but really statistics should be involved in the entire scientific process, starting with data collection, which we'll talk about in this section. So how do we talk about data? Well, we talk about data in terms of observations and variables. So the observations in a data set are the individual subjects or the individual participants in a study. In the health sciences, these are almost always, but not necessarily, the people that we're trying to talk about. The variables are then the individual characteristics that we record for each observation or each subject in our sample. When we store this information, we want to do so in a way that our data is tidy, it's clean, we can access information from it easily. The best way to do that, and the way that most software thinks about data, is to have your observations or your subjects along the rows, variables along the columns. So that's how we'll present data and work with it in this class. So here's an example from the last video, our case study about the peanut allergy. Here's what the data table might look like. Each of these individual rows represents a different person who participated in the peanut allergy study. Each of the columns represents a different variable that we recorded. Now, what type of data we have, what type of variable we have specifically, tells us a little bit about what we can do or what population characteristics we might be interested in. So let's think about how we could categorize these variables. Variables typically fall under the numerical category or the, categorical category. Numerical variables have numerical values with meaning. So the numbers have to mean something relative to one another. A classic example of a variable that has numbers but no meaning are zip codes. It doesn't make sense to add and subtract zip codes. They're just numerical identifiers. So we wouldn't consider something like that a numerical variable. However, something like height, weight, blood pressure, those have relative magnitude. Those numbers have meaning. So those are, in fact, numerical. The other type of variable that we frequently see are categorical variables. So these are, simply put, just categories. What color shirt you're wearing today, that's a categorical variable. Whether or not you're watching this video on your phone or on your laptop or a desktop, that's a categorical variable. Remember, these categories can be represented by numbers, so it is a little tricky sometimes to distinguish. So let's take a look at this data set from the LEAP study. First, identifying the categorical variables. Most of the variables in this data set were categorical. Participant ID, that ID number is categorical because it doesn't really make sense to add and subtract participants in that way. The treatment group is categorical. The sex or gender is categorical, primary ethnicity, categorical, overall outcome, categorical. Our numerical variable in this data set, that's the age at which the participant was enrolled. So how old were they in months? Let's take a look at another data set. So the functional polymorphisms associated with human muscle size and strength study. One thing you'll notice with these health studies, by the way, is some weird capitalization sometimes or some strange word structure, all to get a nice acronym. It's all about the acronym in the health sciences. So this is the FAMOOS study. This study measured a variety of demographic, phenotypic, and genetic characteristics, and we've got 1,300 roughly participants. One study that was done based on this data set took a look at the relationship between muscle strength and genotype at a particular location on the ACTN3 gene. So the idea being that the gene that's expressed at that location might have something to do with how much muscle strength a person can develop. Here's the data. We won't get into that quite yet, but we will later this semester. Let's just talk about the types of variables that we have. So types of variables. The categorical ones are fairly straightforward to infer. We have sex or gender, we have race, ACTN3.R577X, that's the particular gene and location, CC, CT, those are the genotype expressions. Numerical variables, BMI, body mass index, we've got that one, height and weight, age, NDRMCH, DRMCH. We'll talk about what those mean a little bit later when we come back to this data. Now, beyond numerical and categorical, we can think about breaking variables up even further. In the case of numerical variables, how we address them doesn't necessarily change that much based on whether or not they're discrete, so it can only take on a few possibilities, or continuous can take on any possibility within a range. So we won't worry about that distinction too much. However, in the health sciences, we fairly frequently see variables that are ordinal or nominal. So I do want to talk about that a little bit. An ordinal categorical variable is one with order, one that has meaning. So for example, if you ask a patient to rate how they're feeling today, are they feeling the same? Have their symptoms worsened? Have their symptoms improved? There's definitely an order here. Worsening symptoms, same, improvement. That would be ordinal. Variables that are nominal don't really have a natural ordering to them. So eye color, for example. There's no reason to report eye color in a particular order, so that would be a nominal variable. So let's take a look at another example. Suppose that on any given day, a research assistant is collecting data on the first 20 individuals visiting a walk-in clinic. We have age, sex, height, weight, and reason for the visit. Let's classify each of these. Age in this example, how is that reported? It's actually measured in ranges. So we have age as less than 21, 21 to 65, greater than 65. So this is an example where age would actually be a categorical variable, and it's an ordinal categorical variable. There's a definite ordering to reporting age in that way. Sex would be categorical, nominal, there's no ordering there. Height and weight, numerical, continuous, so you can take on any number within a range reporting height and weight. Reason for the visit, that's going to be categorical, nominal. There's no reasonable or agreed upon ordering for that reasoning. So the next thing that we want to talk about when we're thinking about how variables should be analyzed is the role that they're playing. What's the relationship between the variables? Is there a relationship? If there might be a relationship, then we want to assign variables roles to play within that relationship. Typically, we'll break those into response variables and explanatory variables. So response variables, these are the variables of primary interest. These are the ones we care the most about. Think of them like the why for the study. They're what we really want to know. The explanatory variable is a secondary variable that can help us explain changes in the response variable, or may be responsible or causing those changes in the response variable. So for example, if we'd like to know whether or not cancer cells can be selectively targeted using antibodies that look for a specific protein marker found in tumors, we might do something like this. So researchers in this study grafted human cancerous cells onto healthy adult mice. Some of the mice were then injected with this tumor-specific antibody, others were not. The goal of the researchers here is to study the number of mice exhibiting lymph node metastases, so beginning to show some signs of cancer in those two groups. So we want to know what variables we've recorded, and we'll focus on this second question. Which variable is the response? Which one's the explanatory? So, could these cancer cells be selectively targeted by antibodies? That's our major question. The researchers are really looking for how many mice are exhibiting this lymph node metastases, beginning to show signs of cancer. That's what we're looking for. That's the primary outcome. So that's our response variable. Something may be responsible for these changes, or may be explaining those changes. Those changes could be explained by the tumor-specific antibodies that the mice have been injected with. So whether or not they've received those antibodies, that's our explanatory variable. So here are some scenarios, and we'll take a few moments. I'll let you pause the video, or I will encourage you, I should say, to pause the video and try to identify the most reasonable response and explanatory variables on your own. So scenario 1, scenario 2, scenario 3. All right, so let's take a look at these 3 scenarios. Scenario 1, do bluefin tuna from the Atlantic Ocean have particularly high levels of mercury, so much so that they're unsafe for consumption? We're primarily interested in the levels of mercury, so that's our response variable. Scenario 2, for infants predisposed to developing a peanut allergy, is there evidence that introducing peanut proteins early in life is an effective strategy for reducing that risk? The risk of developing the peanut allergy is our primary variable. And finally, scenario 3, does a recently developed drug designed to treat glioblastoma, a form of brain cancer, appear more effective at inducing tumor shrinkage? We are really interested in that tumor shrinkage. That's our primary response. Now let's look at our explanatory variables. For scenario 1, there actually really isn't an explanatory variable. We are measuring those mercury levels, but we're not looking at any maybe cause of those mercury levels, something that could explain that. So we don't necessarily need to have an explanatory variable in this situation. Question 2, the peanut study. Whether or not we've introduced those peanut proteins early in life, that is our explanatory variable. That's what's telling us about whether or not they've developed this allergy. And finally, for scenario 3, does whether or not they've received this new drug explain the tumor shrinkage? So the drug is our explanatory variable. So in the next video, we'll talk a little bit more about how to design these studies, whether to use an experimental or observational study. That's it for this one. Have a good day.